Welcome to my PPT presentation. This is K. Ruchita Kasa from 2nd BSc Biotechnology from DK Government College for Women in Nellore. A special thanks to Dr. T. Suranjan ma'am for giving me this opportunity. Today my topic is PCR. PCR stands for Polymer Chain Reaction. Now let us discuss about PCR. Now see what is a PCR. PCR is a technique used in a lab to make several copies of a particular section of a DNA. PCR is a strong tool that amplifies the even tiniest particle of a DNA. It is an in vitro technique. The function of PCR is similar as a photocopy machine. The PCR is a reaction created by using an instrument is called thermal cycle. The enzyme which used in PCR is called as a DNA tag polymer. The PCR is discovered by Carlos B. Mollis in 1983. Here the picture of Carlos B. Mollis. Three several steps in PCR. First, denaturation. Denaturation occurs at 94 degrees Celsius at 1 minute. Hydrogen bond between the two strands of a DNA are break down and converted into a single strand of a DNA. The single strand acts as a template. The function of denaturation is breakdown of double strand DNA into a single strand. The temperature should be provided from a longer time to separation of the two strands. Second step in PCR is annealing. Annealing occurs at a 54 degrees Celsius to 60 degrees Celsius at a 1 minute. Primer binds to their complementary sequence on the template of a DNA. This serves as a starting point of the synthesis of a DNA. The two separated strands run in an opposite direction. The two primers are forward primers and reverse primers. The third step in PCR is elongation. Elongation is also called as extension. Elongation occurs at 72 degrees Celsius at 2 minutes. DNTPs are added to the 3' end of a primers by the tag polymerase enzyme. DNTP's full form is deoxynucleotide triphosphate. Elongated the DNA in the 5' end to the 3' end direction. Always the primer travel in a 5' end to 3' end direction. These three steps are repeated from 30 to 35 end direction. These three steps are repeated from 30 to 35 times and large number of amplicons are formed. This is a cycling of PCR. In first only I told that there is a cycle which used in PCR instrument as an instrument that is called as a thermal cycler. Now let us see how the PCR will do. What are the steps? First, denaturation, annealing, and third, elongation. Now let us see the applications of PCR. PCR is used in diagnostic of a genetic diseases, used as a tool in a genetic fingerprinting and parental testing to differentiate and know about the relationship between the organism, analysis of gene expression and HIV identification. This is the documentation which is used in PCR for doing the PCR. Like this the PCR will do. Thank you for giving me this opportunity.